ไปถามน้องอื่นก็ได้
ได้เลยถ้าทำอะไรกับเพื่อน
มีปัญหาที่วอลนะ
ได้เลย
เขาไม่อยู่ตรงนี้เพื่อได้แค่เซ็กชันแรกต้องลงไปกดข้างล่างต่อ
แต่นั้นมันบ่อยไม่ใช่หรอเราเบรกพร้อมตอนเช้า
ที่มันออกเลย
พอค่ะ
ไม่ได้ปวดเลยนะแต่มันได้ยิน
เป็นเสียงคนอื่นหรือเปล่า
Good morning
เราเพิ่งเปิดเองลืมเปิด
Good morning, Mr. Masahiro.
Hello, can you hear me?
Yes, loud and clear.
Okay,
Yes,
thank
I
you.
can hear you.
And I think, hi, Poti, and hi, Dr. Kajita-san, and how many people are coming yet? I think we should wait for another 10 minutes or so. Is there any problem? If not, we'll just wait here.
Nakako-san, can I test my sharing the screen?
Yes, it's a nice occasion. Yeah, you can test. I'm not sure. You can try sharing your screen now.
Yes. Okay, thank you very much. I try. Let me see.
I think we need the permissions and unshare before we can share.
It's not permitted yet. I think.
So the admin have to stop sharing before we are. Yes, probably now is okay.
Okay, yeah.
Okay.
We can try.
Thank you very much. Yeah, I can try. Thank you. I try. Yes, I'm now looking at my screen and let me see. This is the one. Yeah, I already have problem for when I start slideshow. Wow, it's very small. I see. So, Nako-san, do you know how I can enlarge the display? Actually, my screen display is for the image size for full screen, but I'm now looking at from my other PC, and the slide is very small. Do you think it's better to use the PDF? Yeah, it works, but how do you think? What is the size of my screen in your PC? Is it small or big? どうですか？なんか
It's full screen now.
Yeah, it is full screen. Yeah, in my PC.
Yeah, for me it's okay.
It's okay.
It's full screen.
I see. All right. No problem. So maybe only for my screen in my PC, look up another one. All right. Thank you very much. I stop my sharing screen.
Maybe you should check your what to say. But because you are sharing the screen, I think for us, just click the share the screen will be enlarge than the full screen.
Anyway, no problem. Thank you very much.
Okay, then I will try to share my screen. Or
mine
is, you can start.
Can you see mine? I think
it's a wide screen.
Yes.
No problem.
So, okay.
Good.
I guess it's because of you have two monitor in front of you.
Yeah,
Yeah, that's why
Hmm.
there are some, the size is different.
maybe. Anyway, no problem.
All right. I will try mine. Excuse me.
Excuse me, Mr. Nagaoka, I'm calling you from admin 2.
Pardon me?
As can you see the friend that say admin 2 on the screen?
I'm now looking for my slides, so please wait
that's
for a while.
okay.
Sorry. Actually, it's duplicated quite a lot of place, so I don't know where is the latest one. Okay, this one, and this one. All right. All right, this is I will share from now. Okay, so you can see my screen. It's now, yeah, it takes time. Presentation mode.
Yeah.
Yes,
Okay,
it's fine.
good, thank you.
So, Nakaoka-san, in the part
Yes.
2 session of this incubator, we have 40 minutes. Then there is
I think for the part 2, each one have a
10?
Time for 10 minutes each.
10 minutes, okay.
Mm.
Then 10 minutes including a question, or we should stop something like 9 minutes or 8 minutes?
I don't want to be so strict about time, but I hope we can try with accepting 1 or 2 questions for each speaker. But if it's too late, maybe we will just going to make it in the last discussion session, or end of the discussion, end of each part.
So I will aim 9 minutes something. Try.
Yes, please try to keep it time, but it's actually, you can, I don't want to, if it's too long, I say something in the middle, but I don't measure time, so don't worry about it.
Thank you very much.
Okay, I just want to confirm whether all the speaker is here or not. If you are a speaker, please change your name in Zoom to show you a speaker, so that not only myself, but our organizer can easily understand who is going to give a talk. Okay.
Mr. Nagaoka, can you hear me?
Yes.
Yes, okay. I'm the admin and I will be hosting the meeting with you today with my colleague. So as you can see now on the screen, it says admin 2.
Yes.
Right. Later on the screen, it will change to timer instead. When the session later on, the screen will change to admin 2. Will change to timer instead once the session starts.
Okay, it's going to be timer. Okay.
Timing, yes. Okay. So my question is, this session divided to 4 parts. would you like me to time it separate in each part, or you want all the time together?
Okay, for the first 6 talks, please set the time to 10 minutes each.
For the first pattern?
For each speaker, please set the time for 10 minutes.
For first speaker, you mean the first part?
First 6, let's see. I think it's okay. You can just set 10 minutes for all the speakers.
10 minutes for each person, right?
Yes, that's true.
Okay, all right.
Thank you.
Thank you very much.
That will be very helpful. Oops.
Recording in progress. Hi, good morning, Masa. Anjana here.
Okay, nice to see you, Anjana.
Yeah, just to check in first. Thank
Okay,
you.
do you want to test your presentation?
Yes, I
you have-
can.
Yes, just a quick, you can try now.
Okay. Okay, can you see it?
Yes, and you have a YouTube.
Great, thank you.
Good, thanks.
Maybe I quickly run YouTube also if nobody here yet.
I think your movie, YouTube.
I have a video.
I
In
hope
case.
it works. Yeah, it depends on the speed. of the
Is it
internet,
okay?
but looks okay. It's moving a little ziggy way, but I think everybody can see it.
Okay, okay,
Right.
okay. Thank you.
You're welcome.
And Mr. Nakaoka, just a short reminding that we will be having a group photo before the session starts.
Do we have something before the session starts?
They would like to have a group photo.
Okay, so do you think it's better to have it in the middle or before the start of the session?
Maybe before would be good, so we don't have to interrupt the speaker later.
Let's see, either is fine. Sometimes people comes late, but I don't think it much matters. So let's try to take a photo first before the session starts, okay?
Yes.
Mm. So wait for 1.5 minutes. Ooh, many people coming. Okay, thank you everyone for coming to the incubation session of Marine Life 2030. Actually, it is one of the starting session of the UNO Decade of Ocean Science WESTPAC kickoff start meeting. We will start our incubation session 2, Marine Life 2030 from now on. And the administrator request us to take group photo before start of the meeting. So if you can, please show up your screen, videos, and show your face, and we will take a photograph now. Okay. How many we are here? We have 30, so maybe we need to screenshot. So, all right, it seems everybody looks ready. If not, please say no. Okay, and wait a little more. It seems okay. All right, then please smile. We'll take several photos because there's two separate screens. Okay, keep smiling. I'll take some photos consecutively. I think administrator will also take a photograph, so it takes a little while. Okay, one more shot.
Okay, thank you very much.
It's okay. I also take a screenshot, but I think organizer took it already.
Yes, already. Thank you.
Okay, that's really good. Thank you very much. I have to swipe before starting. Okay, all right, then we'll start our session. Can I share my screen first? Okay, let's start our session. So welcome again to our session. So Decade Action Incubator 2. It is entitled Marine Life 2030, Increasing Our Knowledge of Marine Biodiversity in Indo-Western Pacific region for better conservation and management. Quite a long title anyway. Okay, so from now on, we have a 2-hour session about focusing on this new program called ML 2030. Marine Life 2030 is actually one of the key program or action plan, which is actually proposed to Ocean Decade of Marine Science, Decade of Ocean Science program in this January. And it was endorsed soon. And it actually consists of quite a large number of organizations, institutions, groups, research groups, and NGOs. I think I couldn't count up how many projects or how many groups are involved here. But we decided to do unite ourselves to do better research and dissemination of marine biodiversity research. This figure shows a brief history how our international effort of marine biodiversity research has been going on. So since the establishment of GOOSE, the Global Ocean Observation Systems, quite a lot of collaboration for unifying the marine observation, including biology and marine biodiversity, has started. And one of the highlights started from 2020. The actually project called Census of Marine Life was conducted over the 10 years until 2010. And there was a lot of scientists involved and a lot of research to clarify the gap in our understanding of marine biodiversity. As a result, there's quite a new discovery of new marine species occurred, and a lot of papers and the new distribution records obtained. And then they are actually launched, and on the database, OBIS is also produced during this era. And actually, our marine biodiversity research has greatly improved. After the Census of Marine Life, we don't have to source as big funding as that before, but we keep, some of us keep collaboration towards a new study. And Apm Bond, Marine Biodiversity Observation Network, it was established in 2014 under the umbrella of GeoBond, Group of Earth Observations Biodiversity Observation Network, with some key institutions like NOAA or NASA. And then Marine Life 2030 started this year. During this era, of course, all of you know, there's a big change in our climate, I mean, marine environment, not only marine environment, but Earth environment. So temperature rises quite a lot in these centuries, especially during our research period. But we also know, we also have understand ocean is a home to many marine species, and it has high economic values. And many people, especially coastal people, rely on not only fish catch, but a lot of jobs related to marine ecosystem services. And one of the missions we need to do is actually develop a new, good, efficient methodology to launch essential variable for observing ocean. GOOSE is developing the essential ocean variables. Some of them are physical and some of them are chemical, and we have some components of biology or ecology aspect. In the meantime, GeoBond has been establishing essential biodiversity variables, EBVs. So EOVs and EBVs somewhat overlapped, and we need to develop an efficient method to integrate and efficiently retrieve and analyze these essential variables. This is my final slide. So we have a requirement now from the decade of ocean science to do more efficient and concentrated research on marine biology or biological oceanography or marine biodiversity research. And there's already an existing platform and organization and network like GOOSE, Apm Bond, and others, which need to facilitate and coordinate for the better observing systems. And then we need to gain more data on ocean biodiversity to filling our gap, especially in South Asian, East Asian, and Southeast Asia, where is a hotspot of marine diversity. And for the synthetic analysis, we give help to build a coherent, consistent, and coordinated ocean observing systems, which will be used for better management and better conservation of marine biodiversity and coastal marine ecosystems. Okay, so in this session, we have another introductory talk by Dr. Leopoldas showing our effort in Asia-Pacific region. And then in the part 2, we will have 4 talks about introducing the cross-cutting edge of the current and ongoing research progress in our region. And finally, in the part 3, we have 2 talks. Actually, it is linking science to society and policy. And finally, if time permit, we want to have a general discussion how to proceed our new program on marine biodiversity. Okay, thank you very much for your attention, and I will pass it to Venus, and we will accept any question after her talk for the introductory part. Okay.
Thank you, Sensei. Let me. Once again, good morning, everyone. It is indeed very important that we have this networking nowadays for researchers. And so I am now going to focus on the contribution of the Asia-Pacific Marine Biodiversity Observation Network. And so the Apm Bond, we call it, the abbreviation of that is one of the regional biodiversity observation networks that is part of the GEO Bond. And there are several thematic bonds, and one of that is the Marine Biodiversity Observation Network. Now, the mission of the Apm Bond is we set 5 missions: the monitoring and data collection, research, technology development, capacity building, and linking science to citizens. And of course, this is focused on Asia-Pacific regions. We have the official launching of the Apm Bond in Kuala Lumpur, Malaysia, last June 2019. And Professor Nakauka is the chair of Apm Bond, in which the Secretary General is Takisha Yamukita. The office is in Jamstick, and we have the website here in the link. You can see the link for the website of the Secretariat of Apm Bond. So one of our contributions is on monitoring. We have this monitoring sites 1,000 coastal program. I actually had one part of this during my dissertation. This is a nationwide program in Japan wherein there is a monitoring of 1,000 important ecosystems over the next 100 years. And it started since 2001. And I was lucky enough to be able to have the data, some of the data for the seagrasses, the macroinvertebrates and the seagrasses in the coast of Japan, and we had it published. Another monitoring efforts that we have for Apm Bond is on the part of the Census of Marine Life, which is one of the projects is the Nagisa. It's focused on the on the shore, on the near shore, soft bottom seagrass and Marco-algal covered rocky shore communities. And there are several achievements for that. Data has been, data on biodiversity has been increased, and this has support a lot of scientists for the geographic distribution and georeferencing of the species records. Another contribution is on the data collection and management. So as you can see, there has been a lot of huge data gaps on understanding the seagrass in around some parts of the Asia Pacific, but we have ongoing contributions for that. In fact, there are already several publications for that. One of the students of Professor Nakauka, PhD students, had published papers with regards to the distribution of the seagrass beds in Southeast Asia, and that could be a very good reference for us. And in terms of research, so one of the regional bonds under GeoBond is the US bond. They have this pool-to-pool project assessing several biodiversity areas in parts of the US, but we also replicate that in parts of the Asia Pacific. And so, in fact, in the part 2 of this session, Dr. Zhao will talk about the big data analysis of marine biodiversity. So please, we have to see his presentation. So we have this ongoing research on the quantification of blue carbon in Southeast Asia. In fact, there is already one very good publication of that. Dr. Pratik will talk about the blue carbon study, the part 2 of this session. So aside from the research and the monitoring, we are also into the development of new technology. And so one of that is on the using the remote sensing and remote sensing, GIS, and doing the species using the distribution model, the deep learning mga techniques in order to forecast or understand the fate of our ecosystems from using data from the past and the current, and forecasting the fate of that areas in the next coming years. And we are also developing the eDNA methodology in the assessment of the biodiversity of our important ecosystems. There is Dr. Kajita already have this research on the mangroves, and Dr. Kajita and Dr. Lim will talk about eDNA metabolic coding studies in the part 2 of this session. That would be very interesting. So another thing that Apm Bond wants to contribute is on the capacity building. We have actually this ongoing planning regional workshops on marine biodiversity researches in Asian countries. And there is also networking international summer courses for graduate and undergraduate students. And this has been taking place for several years now in Hokkaido University under the laboratory of Professor Masa. And you can see the schedule there. And there is also a schedule that starts for 2022. And I added here, there is an ongoing eDNA methodology online training. And actually it was done, I think, last week by Professor Kajita. And there will be more to come to involve the young career professionals on the eDNA metabolic coding if our Asia proposal will be approved, hopefully next year. So the proposal, I will talk about more in the proposal on the next slide. And more importantly, we want to link science to citizens. And so studying how changes in the coastal ecosystem affect human society. And so we involve our local partners in order for the science to be brought to the people. And there's also co-design, co-production, co-delivery with local stakeholders and citizens. And more importantly, develop policy recommendations for the decision makers. Apm Bond developed seagrass patterns, wherein our citizens can use it to pinpoint where the seagrasses are, and this will really help in the quantification later on for our blue carbon ecosystems. And there are also summary for policymakers, wherein it can be used for sustainable uses of marine ecosystem services in Japan. So I guess there will be more applications to come so that we can probably laymanize our science to the people, and they can really help us in improving the understanding of science. And another thing is on the studies linking coastal ecosystem services to local communities. This is a kind of very important research wherein you involve people to understand their vulnerability and well-being, and with regards to ecosystem services. And the paper of Ma'am Keros is already in preparation. And with regards to the cultural ecosystem services by mangrove and seagrass bed, a source of happiness. This would be a very interesting paper, a very good reference. So Dr. Porwante, Dr. Keros, and Dr. Fortes will actually talk about transdisciplinary approach to bridge scientific knowledge to human society in the part 3 of this session, as what Masa mentioned a while ago. So some steps forward. So with the leadership of Professor Nakauka, the GSPS Kakini project, it was actually approved already, but because of the pandemic, the implementation of this project will be started in 2022. Please correct me if I'm wrong, Masa. And so it's with regards to the evaluation of our important ecosystems, particularly our seagrass. So there will be forecasting on what will happen to our seagrasses in the near future. And another thing is we have this, I mentioned a while ago, we submitted an eAsia proposal for eDNA. So this will be focusing on the Indo-Pacific under the leadership of Professor Kajita. And just news from the Philippines, our proposal from the Philippines will be under the evaluation of the DOST by next week. So hopefully it will receive a very good approval, so such that it will be approved next year. So we will be assessing several mangroves in the Philippines, just getting the water samples, and then do genetic analysis in order to determine what are the species are there, and we will do it in the next coming years so that there will be monitoring of the biodiversity. So more or less, we will already be using the eDNA more advanced methodology for biodiversity assessment. So Apm Bond had already, we already contributed a lot for the advancement of our research, not only within the scientific community, but also for the locals and decision makers to be to use. And we are still asking for more members so that we can expand our network. So you can register to Apm Bond. There is an email there wherein you can just contact to be able to register. So that would be all. Thank you very much.
Thank you, Venus, for nice presentation about our activity in Asia Pacific. Before moving to part 2, I'd like to welcome any one or two quick questions or comments, or you can, of course, leave your comments or questions to the chat box. Is it? Or you can raise your hand, but sometimes if it's already more than 60 people, so sometimes we overlook even if you are raising your hand. Okay. If no questions, can we move to the part 2, cross-cutting edge research in Asia regions, right? All right, then I first welcome Dr. Quincio Zhao from Ocean University of China. He will talk about big data analysis in global scale analysis and the implications on Southeast Asian sessions. Okay, Quincio, your turn.
Thank you, Masa. Can you hear me?
Yes, clearly, thank you. Yeah, you can, we can hear you.
Okay, thank you. Okay. Yeah, I'm going to share my screen. Can you share my slide?
Yes, it's clearly,
okay.
Yes.
Okay, thank you. Yeah, Yeah, okay, good morning, everyone. I'm Zhao Qianshuo from the Ocean University of China. Today, I will talk about a research topic I was and I'm working with, prioritizing areas for marine biodiversity conservation from global to regional. So it was in 2016, the IUCN World Conservation Congress called for the protection of at least 30% of each marine habitat globally and 30% of all the ocean for an effective marine biodiversity conservation on a global scale before the year 2030. This call was repeated in the COP 15th conference just this October in Kunming. It was still a small number in 2020, and so it is still far away from the 30% goal before the year 2030. To respond to the call, I and my collaborators did a research on a global scale, try to find the areas where would best represent 30% of ocean biodiversity. In this research, we included the factors of marine ecosystems, seabed topographic variation, species richness, marine biomes, and species ethnicity as realms. This paper was granted the Elsewhere Atlas Award for its contribution to the United Nations Sustainable Development Goal number 14, the biodiversity conservation underwater. The ecosystems layers were derived from previous research of mine. The ecosystems mapping was based on an unsupervised cluster analysis of 20 physical, biochemical, and nutrient variables. Some areas belonging to the same ecosystem were geographically divided by continents, but distributed at similar latitudes and symmetrically on both sides of the equator. The marine biomes contain the seagrass, kelp, mangrove, and shallow water coral reefs. In this research, biomes were defined as large geographic areas characterized by the same plant life form, which form large areas of three-dimensional habitats with high primary productivity. Specifically, the shallow water coral reefs was defined as biomes because they host photosynthetic algae and provide complex habitat structure. The seabed topographic variation, the rugosity, was based on the depth data. And so it also covered the benthic geographic features such as canyons, seamounts, and abyssal hills. The layer of within realm species richness was compiled by the data of species richness and species ethnicity together. The purpose here is to know the condition of species richness within each species ethnicity realm through comparing the species richness numbers within the same realm rather than the ones from other realms. After all the layers were ready, they were introduced into the software zonation. The basic algorithm of the zonation is to iteratively remove cells from the lowest significant cells up to the highest ones. In this research, it's used the rule of target-based function. The TBF method prioritizes the cell which can contain the presence from multiple layers rather than a cell which only cover an extremely high cell value from a single layer. To satisfy the purpose of this research, all the layers was set target of 30%, which is the 30% of each layer is expected to be covered by highly prioritized areas. So this is the prioritization result of the research. The prioritized areas were mainly located at the continental coasts, island arcs, oceanic islands, the southwest Indian Ridge, the northern mid-Atlantic Ridge, the Kerr Triangle, Caribbean Sea, and Arctic archipelago. This figure is to evaluate how the prioritized areas covered the layers used. The prioritized areas generally achieved 30% coverage on presence-absence layers and fairly covered the top 30% of globally covered layers. In 2017, Assad concluded ecological criteria for biodiversity conservation. This research covered 6 criteria for the importance for life history stages. In future work, it could add the important breeding and growth areas for species threatened with extinction. There is a recently published paper considered the criteria, and for the ecological integrity, the human impact factors could be added in future work. This slide shows that the prioritized areas were only covered by a small number of MPAs. So countries could plan more MPAs in their own EEZs to for the biodiversity conservation. So this is the paper I just mentioned. It is a recently accepted paper which added threatened species to represent biodiversity areas. I am a co-author of this paper, and Tamlin Jefferson is the corresponding author. He found that if to protect 30% of the range of each threatened species, we need to expand the necessary percentage to expand the coverage of the protected areas to 30% of to 40% of all the ocean. In further, I analyzed the 5 data-driven marine prioritizations, which were based on different aspects of biodiversity conservation. So from the consensus map in this slide, the red areas in red circles represent the locations where were prioritized by 4 or more prioritizations. So if people want to start marine biodiversity on a global scale, they can simply try to plan MPAs from the red areas. Based on the idea and the method for biodiversity for marine biodiversity conservation on global scale, I and my collaborators participated in a proposed project for the biodiversity conservation in Europe seas. This proposed project was led by Mark Costello from the Nord University. In this project, I initially did an analysis to identify the distribution of different kinds of ecosystems. And meanwhile, there are also available open source marine species distribution data from the OBIS Europe. And so then I did an initial prioritization analysis for the method verification for the proposed project. And the figure on the right-hand side was added in the proposal main text to demonstrate what the main output of the project would be like. In further, I and my collaborators try to do a similar project in the West Pacific from the very north to the very south. So this time we would like to try to do the work with the ecosystems on surface and near bottom together. So the figures here show the distribution of the surface ecosystems and benthic ecosystems. So we also collected the species richness data from the open source data site Apm Maps. And with the layers just shown, we did an initial prioritization analysis for the project. And in the testing, we added the layers of ecosystems, species richness, spread topographic variation, and the marine biomes. And we found something interesting, and we would further analyze the things we found from the work. And thank you very much.
Thank you, Francois, for nice, really remarkable analysis at the global scale. And I'm really look forward to your Asia regional analysis from now on. Okay. And time is passing, but if you have any one or two quick questions, you can ask right now. So, Dr. Kajita?
Yes. So thank you very much, Dr. Xiang, for very impressive presentation. So I'm really interested in the global research for, especially for biodiversity, species richness. Then according to your data, I found that you have maybe included the analysis for mangrove ecosystem, but I wonder, how did you get the species richness data for mangroves? So I'm wondering, what is the data sources for the species richness of mangrove?
The distribution of mangroves were also derived from an open source data set. The WCMCUNEP data set, I think it is, the data set was called Ocean Data View. Similar to the name of another software, but it is called the Ocean Data View. And we collected the distribution of mangrove from that data set.
So that's the obvious data
on global scale.
So that the obvious data included even not only for the fish data, but also for the benthic data as well?
There are benthic marine species occurrence records from the obvious data. There are.
Okay, thank you very much. I should explore more for the obvious data. Thank you very much.
Thank you.
Thank you very much for a nice discussion. We now need to move to the next speaker, Dr. Anchana Prathep, talking about blue carbon. Kianshu, could you release and share the screen? Okay.
Okay.
Yes, Anchana, it's your turn.
Okay. Good morning, everyone. Just to checking first, everything is fine. So very good morning, everyone. Thank you, Professor Masa, for the opportunity. So today I would like to talk about the blue carbon. And with this topic, actually have to thanks for the network of the WESTPAC. We are able to gather this kind of information because there is this kind of platform for us to really share the information. So let me have a quick recap about what I'm going to talk about. We are doing this blue carbon in the seagrass. We are trying to understand how much the carbon could be installed. If you see this, the quick recap is if everything going well, we are conserve our seagrass. The seagrass would be a very good natural source to sink the carbon. It can help mitigate the carbon, which is a problem nowadays. But if we do nothing, the carbon that actually trapped within the seagrass ecosystem, it will be released back to the atmosphere. So it's our turn to really understand and try our best to be part of this combat with the climate change. I wouldn't go into the detail at the moment, but if you are interested, you can really go and see this further information from the publication that we recently published with all the network. Now, let me introduce you further into the blue carbon world. We have been working on the blue carbon for some time. We are able to get some kind of information, and we think the best way is to do it is to share it in terms of the video. So allow me to share the video for you to have a good recap and understanding of this blue carbon in a very nice, easy way that we can communicate to the other through this video. Okay, I hope you can see it. Okay, so you can kind of have some idea what actually we've been doing, and then be able to digest something to easy for everyone to really understand about what's going on in this seagrass ecosystem. But if we look at it, actually nowadays is an opportunity for us to really highlight the seagrass ecosystem using blue carbon as another umbrella for us to work on. Now, what we have is actually this kind of collaboration that we work together using this kind of platform. If you look at this map, right, we have some of the seagrass data. But if we look closer, actually, there are still a lot of holes that we like to fill in, strengthen our collaboration, and using what we already developed from previous study to make the module is much more verified, much more be able to disseminate this kind of information to the others. This is one of the data points that we've been working and collecting from our previous collaboration. This is another example, actually, by the leading of Dr. Sudha, right? We are able to really understand what actually going on with this temporal change of the seagrass in this kind of region. So let me allow me to really go into this kind of conceptualized model. What we do with this seagrass, actually, we know that each country have different capacity to go out there and collect the data. We try to simplify our model. The starting point is just to look into the percent coverage, and then we are going further, develop some of the model that we are be able to understand the above ground biomass, the below ground biomass. But if you go out there and you already have this kind of information, that would be great. That would make the model much more stronger. But start from the coverage. Of course, we are able to really go down into the carbon in the sediment, which is where it is the hugest amount of the carbon that's stored in the seagrass ecosystem. By having this simple model, right, at the end, we are able to really estimate the carbon in the vegetation and then the carbon that's stored in the sediment, which is the heart of this blue carbon. What next we do? We have this kind of number, and then the next step is we really have to go out there and then estimate the seagrass area in each country. That's when we come to this kind of number in our study, just to simplify you and have a good understand that actually everyone can help to fulfill the gap, right? Just to show you some of the information we have, which is quite surprise for us. We found out that actually the mixed seagrass had the number of carbon storage lower than the monospecific seagrass. So this kind of the trend, but actually if we look at certain part of the world, Indonesia, maybe they act differently. Malaysia is not really clear cut. And if we look at this error bar, it's still very high. So definitely it's very important that with kind of this project, we really have to work together. Again, look back into this. The starting point is just we need the coverage. If we have much more point to simulate this model, that would make the number of predictions stronger. Give you some further information, right? The number of the carbon storage, probably Philippines would be the most carbon storage in the region, right? Look closer. The number of the carbon actually they storage in the sediment more than the living biomass. But don't forget about it. The living biomass is actually also very important because they provide another ecosystem service to the livelihood of the people and to the healthy ocean there. Look closer, what else we have? If you look further where we are in the world here, the number of, let me go back here. The number of actually what we have in the Southeast Asia is quite high in comparison to the other part of the world, as we know that we are hotspot of the marine biodiversity. So the blue carbon could be just one to add on the highlight of this most important ocean. So how are we going to collaborate and help to fulfill all these gaps with the blue carbon? It's just simple thing. This is the data sheet that we actually sent out to our collaborator. What we have is actually simple things. If you already have the data, species present, the substrate, the coverage, and if you have the above ground, below ground, that would be great. The time of your collection, the location, and then the other information that you have. If you are interested to really provide us with some information, you can actually scan this barcode, and then you would have this data sheet. The simple things that we can do is just hopefully that we can estimate the amount of carbon that storage in your seagrass ecosystem for you. It's probably not that exact, but actually this is a good starting for the further collaboration. Just to give you how we can collaborate on this blue carbon. Now, what next we like to do? I think it is important to also understand where the carbon comes from. Some of our studies actually show that the carbon that's stored in the seagrass, some actually come from the mangrove. And then also some other study showing that the carbon that's stored in the seagrass come also from the coral reef. As we all know, all these ecosystems, they connected. So it would be interesting if we can kind of having this kind of collaboration to really see where the source come from, how much it contribute in each ecosystem. So that would be a good information for us to really understand and be able to manage or conserve our ecosystem effectively. What else we can do? We know that nowadays, if we look, this is some of the examples from Thailand. Healthy seagrass we found in 2016. It is having something wrong with it. It being disturbed. By 2021, the number of seagrass decreased. Go back to the first slide that I mentioned. If we cannot keep the seagrass, the carbon will release back into the atmosphere. So this is something that we really have to be concerned in terms of conservation and how to keep the cap of the carbon storage. Okay, once it happens, what should we do? We can really help with the restoration. There's a lot of restoration ongoing, but we don't really have a good understanding in terms of how much the restoration actually can help keeping or trapping the carbon back into their ecosystem. So this is something that probably we can really help work together and share the idea. And then, of course, at the end of the day, we really hope that as a seagrass ecosystem, we can really help tapping the blue carbon into this ocean, and working together is very important. The blue carbon actually is a very good strategy for us at the moment because it actually helps to people look into this ecosystem and help to preserve. We still have a lot that we can do together in terms of marine biodiversity, but blue carbon is just part of it. Okay, that's all I'd like to share. Thank you.
Thank you very much, Antenna, for nice presentation. And the YouTube video is very helpful to help citizens to understand how important blue carbon ecosystems are. Okay, I'm sorry, but the time is running up. So if any of you audience has questions, please write it in chat, and Antenna can answer later on chat or by in the general discussion time. Okay.
Okay. Thank you.
Thank you again, Antenna. Okay, we move on to the next talk by Dr. Kajita about eDNA. So, Dr. Kajita, you can start when you are ready.
Okay, can you hear me? Okay. Thank you very much for introducing me and also giving me this chance to introduce our project on eDNA metabolic coding network in mangrove ecosystem in Asia. So I'm Tadashi Kajita from University of Ryukyu, Japan. So as you know, mangroves distribute pantropically. That means in the coastal region of all over the world in tropics and especially in the subtropical to tropical zones. Then it has occupied in 123 tropical and subtropical nations and territories. And the largest mangrove area is in the Southeast Asia. Then, because of course it is in between the terrestrial and the
ドクター梶田、you are muted.
So sorry about that. All right, restart. So I'm Tadashi Kajita from University of Ryukyu, and I'm going to talk about developing eDNA monitoring network in mangroves ecosystem in Asia. Okay. So the mangrove are distributed pantropically. That means in the tropical and subtropical area all over the world in the coastal region. And it is possessed by 123 tropical and subtropical nations. Then the mangrove, largest mangrove area in the Southeast Asia. Then the mangrove is in between the terrestrial and oceanic ecosystem, but we are quite happy that we could join to the Marine Life 2030. Then in the logo, it is also placed in the very important part of the beautiful logo. Then people are in the, especially in the subtropical and tropical region, receive a lot of benefit from mangroves, from the wood livelihood, mangrove ecosystem service, coastal protection, water filtration, a lot. But mangroves are also under threat because of many drivers, especially for the human activities. And the most important one is the land use change by logging and also making farms and so on. Then we know that almost 35 to more than 50% of the mangrove area has already destroyed. And also the degradation is going on. Then the biodiversity in mangrove ecosystem is quite rich, as we know, but the biodiversity is also changing, and degraded and lost. So this is the most important question that we have. But according to the references, so we do not know a lot about the species diversity of mangroves. So you see in this table, so many ND for the mangrove area for many groups of the organisms. So that this is the reason I asked for Kinsha's presentation, that how he could get a lot of species richness data from Orbis. So I really should have to work more on Orbis data. Okay. Anyway, for this reason, we wanted to know, or at least start to document the biological diversity of mangrove ecosystem. But the workload and labor of doing field works in that wide region of mangrove is quite difficult. So that we started to use DNA metabolic coding to study biological diversity of mangrove ecosystem. So this is a simple explanation of DNA metabolic code. As you know, barcode is on the many goods in the shop as to specify what is that shop goods and where it is from and what is the price. Then DNA metabolic code is a very small portion of DNA with only ATGC nucleotide of the sequences. Then we can identify the presence of these DNA within water. Then by identifying its sequence, we can know what is in this water. I mean, what is means which species are in the water. So the okay, this is the explanation. He is Dr. Mia, who established the MyFish primer to study fish species. So when we do the field work, it is quite simple that we collect the water sample from the ocean or the river. Then we filtrate the water to the staybags by pumping it by using a syringe in the field. So this is the basic part of the field work. Then the sample is brought to a laboratory and we extract DNA. But please remember, at this time, the DNA includes everything, every organism in this field. That means from microbe, fish, crustacean, and sometimes insect, many species. But to study that, we use PCR, so using the specific primer to amplify, in this case, only fish DNA. Okay. Then we identify its sequencing by next-gen sequencer, which is a very high throughput machine to generate the sequences. Then by comparing the obtained data with a reference database, we can identify which species, or which fish species in this case, is was in this water. So this is really non-invasive method with excellent maneuverability and simplicity and speed and economy. So using this method, we aim to, actually our ambitions is to study the biological diversity of mangrove, especially starting from fish and crustacean and all other organisms, but on a global scale. But to start that, we have to create a good research network. So we started this one, and luckily we can get the support from JSP's Core to Core program. Then we started this project with colleagues from 9 nations, and actually most people have already joined to this project. Okay. Then in this network and program, not only to do the research, but we try to establish good research network and also disseminate the methodology to the countries, those countries, so that especially for each country, we hope that they can start this kind of project involving many young researchers, then to study the biological diversity of their own country using this method, so that we can rapidly understand the biological diversity of the mangrove on a global scale. All right. Then right now we are obtaining the fund from JSP's Core to Core. And luckily in Malaysia, our colleague, Alison Kim Shan-Wee of Nottingham University obtained a 3-year grant from both CRRP and FRGS. And we also get another JSP's Kakenhi to support this program. In this case, not only for the country covered by the JSP's Core to Core, but we now can expand this research network to other parts of the world, especially to the New World. Then also in collaboration with Nakaoka-san and also Venus Leopardus, we are now proposing to other grant, especially for the Asia, which was introduced by Venus herself. Okay. Anyway, so this is the one picture that very recent one, maybe two days ago, done by Alison Kim Shan-Wee in Malaysia, Selangor. So I said the filtration and sampling is quite easy for mangrove, but for the case of Malaysia, it was not quite easy because of the turbid water. So quite a lot of sediment was disturbed for filtration. So this is a summary, but before going to the summary, I'd like to show you some result, how powerful this is. So just by 1 day sampling in a mangrove river system in Iriomote, in Nakaro River, we could identify more than 100 species of fish like this. And also in a small river, Udara or Yutun River, by collecting water, we could identify some 60 to 80 species of crustacean species, shrimp and crabs like this. Anyway, so this is the end of my talk. So this is the acknowledgement for the all collaborator and the fund. So thank you very much for listening to me. Thank you.
Thank you, Kazuta, for nice introductory talk about eDNA project. And you're getting a lot of good collaboration and data already. Okay, I can, there's a lot of people who wants to ask him any questions. Only maybe I have time for one people so far who's raising hand. Dr. Fortes?
So you're calling me?
Oh, yes.
Okay, thank you. I'm just curious to Kajita, your presentation is really very interesting. Besides the fact that it's new methodology, but I'm just curious about how do you differentiate the genetic materials coming from the mangrove itself, I mean autochthonous, from those coming from the outside or allochthonous?
So,
thank you.
So you mean that the species detected by this method is only from mangrove or from the other area, you mean?
No, I mean, you're working on the water in the mangroves.
Yes.
But we cannot, are you sure that the water is really from the mangrove itself? Because, you know, there is allochthonous coming from the outside, even coming from the coral reef, from the citrus bed. And then also there are those materials which are produced in the mangrove itself. Or is it a part of your research, or how do you control this interchange of genetic materials?
Yeah, good question. And we're not caring so much about that part because we do not make a border between the ecosystem for only the mangrove or others. And of course, the mangrove would be affected by the tide, so that in the high tide, the water in the ocean will come getting into the mangrove. And that time, we have more sea fish and sea species detected in the mangrove area. Then about the area, the method can detect some kind of in 30 minutes, according to one study, the eDNA detected by this method can be something in the area of several hundred meter range. Then the presence of the species or the remaining of the species might be within 30 minutes to 1 hour. So that I think we cannot make a very strict border, but we can say that the species data are from roughly from mangrove.
Okay. Thank you very much.
Thank you very much for your question.
Sensei, sorry, you are muted.
Oh, sorry. Okay, so time is up. So I'm sorry. I'm pretty sure most of you have any questions on this new technique. So please write in your chat box for the question to Dr. Kazuta. Okay, and we move on to the next talk by Dr. Potin Lim. His talk is also related to metabolic coding, but it is for the coral reef health research. Okay, you can start anytime. Potin.
Yeah, thank you. Thank you, Masa. Can you hear me well?
Yes, yes, we can hear you well. You can.
Yes,
screen is clear.
Yes. Thank you very much, and thank you for the organizer for giving me the time to share some of the ongoing work related to coral reef ecosystem and how we actually use the molecular approach using metabolic coding to assess the coral symbiont in coral species that found in 2 islands in Malaysia, Perhentian and Redang Islands. So these are the island are protected area within the marine park of Malaysia. So I think I don't have to go into detail about this because you know the importance of coral reef, social and economically to the livelihood of peoples in this region. And since we are inside the coral triangular area and the high diversity of coral with more than estimated 500 species in this region, and more than 60% or more in according to Veron. So again, that indicate the importance of coral reef ecosystem to all of us in this region. I think, and at the same time, we're also aware that the climate change and other local stressors, for example, eutrophications, other human activity, habitat destructions, overfishing, that have been contributed to the degradations of coral reef. I think that is also very, very clear, and a lot of data and people already indicated that. So, and to investigate the coral reef ecosystem health, I think one of the very important component is to look at the coral symbiont. And with conventional method, it might not be easy to see the changes, how coral species actually experience bleaching that lead to mass mortality of coral hosts, and potentially lead to the collapse of the coral reef ecosystem. I think in this presentation, I will try to explain the process that we have gone through and some of the ongoing work that related to this coral symbiont. And these are some of the facts related to coral symbiont. It's a group of dinoflagellate, single cell, so autotrophic, photosynthetic, and they have various type of pigment that play important role to contribute to the nutritious requirement of corals. So based on the work previously by Ruinis in 2009, that we already know that the coral symbiont actually changed due to various degree of stress, and the coral symbiont compositions also change during the recovery phase, whether it's short-term and long-term recovery. So understanding the mechanistic process of these changes is very, very important, because we know that the coral symbiont actually have different degree of adaptations and different degree of resilience towards the climate, especially the temperatures elevations. So based on the similar work by the same group of researchers, you can see here that now we are referring to the coral symbiont into different clade representing by different genus, Cladocopium, the C, and the D, the Durusdinium, that is very common in highly tolerant or thermotolerant coral species, and the E actually mainly in the free living non-symbiotic coral symbiont. So based on this upgrading, we are able to use metabolic coding approach to assess the coral symbiont in a coral species that found in 2 islands in Malaysia. So our purpose is to investigate the symbiont compositions and to understand the resilience and susceptibility of the coral reef, because we know that the coral reef ecosystem are not made up of a single coral species, but a diverse group of coral. So, and the coral ecosystem health, in order for us to understand, we have to assess in totals to look at all the coral species that found in the coral reef ecosystem. So we're using 2 different islands, the Perhentian Island Marine Park and the Redang Island Marine Park in this study. And this is a island that experienced monsoonal drivers because of the northeast and southwest monsoon, like others island in this region. So we have high precipitations during the northeast monsoon, which is now, and that is the time also for the marine park is actually closed for all the leisure activities. So very small fragment of sample were collected. Then in total in this study, we actually analyzed 65 samples from Perhentian and Redang based on the ITS nuclear ribosomal gene, ITS2 region. So, and I will keep it short, because if you like to know more about the methods that we apply here, you can actually write to us. And my student is also in these sessions that Liket will be able to help to answer some of the questions. So then we perform the Illumina MySeq platform, And then the data will be subjected to analysis using a SymPortal. This is a good Coral Symbiont database that we are able to access. Look at you can have a look at the SymPortals org, and by using this this database, we are able to come out to determine the DIV, the defining intragenomic variant, and at the same time to instead of OTU, we are using the ITS type profile to represent different Coral Symbiont. So from these samples, from the 65 samples, and we actually have 21 general, and with more than 4 million reads were obtained with average 292. And after the sequence filtering, we have roughly about 2.6 million read, almost 40,000 sequence per coral after sequence filtering, and 58,000 unique symbiont DNA sequence post-taxa identifications. From all these sequences, we are able to identify 422 DIV, which is something unique across the 6 general Symbiodinium, Breviolum, the B, the C, and the Durusdinium, okay, the D, Cladocopium, the C, and Fugasium, the F, and the G as well. So, but majority of them actually come from the C and D, which is not surprising for coral. And the coral were populated by 32 ITS2 type of C and D, with 19 C and 13 D. Okay, so, and also at the same time, we are using the cytochrome oxidase to ID the coral species to confirm the coral ID, and they are divided into two main groups called the complex and the robust, based on, of course, also depend on also look at the general morphologies. So all these 65 samples were identified based on both morphology and molecular data to both the complex and robust, and with different family and genus. Okay, so based on the Symbionium composition, we can look at 3 different levels. If you divide them into the figure A, that simply can divide into the group C and D, more than 90% of them. And if you divide into ITS2 sequences, then you can see that the majority of them are for complex is actually the C, and you have more D in some group of the robust group. So, but there are actually some species, some genus, for example, Galaxias, and that actually have a mix of both C and D with more or less the equal numbers. So I think that is also something very, very useful for us to look at them in details, for us to tell which coral species are able to respond well under the temperatures, elevations conditions. So I would try to skip some of this because the time constraint. And you can see here, these are the species richness among the coral symbiont, among the coral families. And that symbiotic will enhance in terms of the resilience. And a lot of study actually look at this. Fast growing normally is more susceptible to temperatures, more susceptible to bleaching. And our study also show that not all the coral species with robust actually host mainly from the D, which explain the thermal resilient mechanism of some of these more robust species. So this is the general thermal tolerance for the coral species that we have identified. The one more susceptible and the one more resilient based on the coral symbiont analysis. Okay, so we actually hope that this coral symbiont diversity analysis can be used in the implementations of MPA in the effort to do coral populations and so on. So, and this is certainly a useful markers for us to look at that in detail, not only in Perhentian, but also in other marine park and other coral reef ecosystem to reaffirm our finding. I think this is a very, very good start and very useful data set. I'm happy to share some of the, if there's any needs for us to clarify further on this. Thank you very much.
Thank you very much, Dr. Potindi. And there's some time to ask for the questions. Does anyone want to ask him any questions on metabolic coding approach for the coral reef research? It is quite interesting. Again, Dr. Kajita-san.
Yes. So thank you very much for Lim for very impressive presentation. So I'm very interested in your method of detecting DIV, in this case, the variation in the genomic sequences. Then could you detect the DIV from the white bleached coral? If it is, we can know which strain is more tolerated or affected by the coral bleaching. Okay, thank you very much.
Okay, so I think the sample that we are working on for these 65 samples, we are working mainly on the healthy corals in order for us to assess the potentials, resilience. We also have similar work by colleague from UMT that were working on the same species but at different stage of bleaching. I guess that will help us to understand the composition changes and also the remaining, coral symbiont that in the bleach samples that can withstand the higher temperatures. I guess for in these presentations, because our intention is to assess the resilience and health of coral in healthy corals. Are they prepared for temperature evaluation or not? So that's why we are not including any species that's exposed to high temperatures or bleach. So I don't think I can answer you at that stage.
No,
so
good answer. Yeah. Understand. Thank you very much.
Okay. All right. Then if you think of any questions, please write in the chat box and each speaker will answer directly or later. Okay. Thank you very much. Is anyone speaking? Okay. Okay. Thank you very much for all the speakers of the part 2. All of the talks are very interesting and it's stimulating for planning our future research project. Okay. All right. We now move to the part 3. It is a session on linking science to local community or policy makers. And we have 2 talks. And the first one is actually co-authored by Dr. Frida Bruanki and Dr. Angela Kiros. And I don't know, I forgot which of you started talk, but now the floor is yours, so you can start anytime. Angela, are you speaking?
Okay, sorry.
I can hear.
Okay, I'm Frida from Diponegoro University, Indonesia. In collaboration with Angela from Hokkaido University, we're presenting our study that focus on coastal community well-being from the Blue Carbon Cultural Ecosystem Service. This is a part of a collaboration between Japan, the Philippines, and Indonesia that we call Blue Cares, JICA and SD-funded program. Okay, next please. Okay, and I will start. Can I control? Okay, yeah, as we know that most population in the world is living in the coast, that is about 60%, and the coast resources also have benefit to the human well-being for their multi-purpose, such as fisheries and also tourism. And the coastal resources also support the coastal community, include mangrove and seagrass ecosystem. That is the blue carbon ecosystem. Okay. Yes, we see that blue carbon ecosystem is focused in the coral triangle here, Indonesia and Philippines, which is this is as a case study, and the hotspot of the country here. The Philippines has high mangrove and seagrass diversity with 40 species of mangrove and 16 of seagrass, while Indonesia has 80 species of mangrove and 12 of seagrass. So see from this tab. And now we talk about the socio-ecological system. This interaction between human and ecosystem that will give service from the ecosystem to human from their action or intervention. And ecosystem service and human well-being is provide a framework that help us to understand the complex benefit and feedback input socio and ecological system. This project is aimed to show how ecosystem service from seagrass and mangrove will contribute to your human well-being. Next, sorry. Yeah. According to the Millennium ecosystem assessment, we see that ecosystem service is categorized in provisioning, regulating, culture, and other supporting. And we will focus for the cultural ecosystem service here that benefit to your human well-being from the element of this good social relationship. Next, please. Please study here, sorry, for the ecosystem service for culture. We call it non-material value that we can get from the spiritual enrichment, cognitive development, and reflection of recreation, aesthetic experience. And this ecosystem service for culture is have less attention than other. And next. Okay. Here the ecosystem service from the culture. We can see from the narrow definition, the subjective component of heart is difficult to measure because we know that this is an interrelation between people to the ecosystem. So among the stakeholders is a complex factor that we have to know, such as knowledge, place of attachment, interaction with nature. And the human well-being is a state that where one can enjoy or satisfaction from the quality of their life in material, physical, and psychological relationship. So people have connection to the ecosystem that contribute to the life for their living. This is what we call it human well-being. They receive the ecosystem benefit from there. Okay, next please. Okay. This cultural ecosystem service, is it a source of happiness? Yeah. People engage with nature as a connection to a place, as a therapeutic value of nature. So well-being indicator from the cultural ecosystem service may also be a source of happiness for local people. Sorry. Okay. Next, please. The sociocultural value is a place that we know a complex sense, such as knowledge here, interaction with nature, and also a place of attachment. Furthermore, next, study found that local stakeholder with a longer time of residence near protected area is put more value on the ecosystem service provided. Okay, and another study found that stakeholder whose well-being is the most dependent on the ecosystem service as are more vulnerable. Okay. And for this study, we try to answer this question: what sort of the cultural well-being indicator do seagrass and mangrove ecosystem provide? Are there differences among group of stakeholders residing in coastal community, from resource user, policymaker, tourist worker, and also other, we call it in the miscellaneous community member. And also with the last, is there any difference across country, Indonesia and Philippines, and also across habitat, mangrove and seagrass? As we focus on study in Philippines and Indonesia, here the two islands we found for our study, we focus on the Busuanga, Philippines, and also Karimun Jawa, Indonesia. Both islands have coastal fishing community and developing tourist industry relatively pristine to the blue carbon ecosystem. So here we have such a number of respondents. And for Karimun Jawa Island is an archipelago in the north of Java Sea, Indonesia. They composed of 27 islands, in which 22 islands is within the national park. And the big island is Karimun Jawa and Kemujan. The park is designed to protect a variety of ecosystems from low-lying rainforest to the coral reef. They have altitude from 0 to 506 above sea level. And the population number is about 9,700 in 2019. And mostly of the residents rely on fisheries and tourist sector. Our study in Karimun Jawa, we found in the two villages here, Karimun Jawa and Kemujan, in which Karimun Jawa mostly from urban area for tourist investment, and this tourist is be the livelihood for the source of income. So the problem there, we found a fish pond development near the Kemujan here, and also they have a low mangrove ecosystem but high seagrass ecosystem. While for the Kemujan, mostly people there is dependent on the fisheries. They have a high mangrove diversity and also seagrass diversity, and the residents mostly have alternative occupation as a gleaner. And yeah, with a problem from the. For the next slide will be presented by Angela. Please, your screen, Angela. Is still mute.
Angela, I can't hear your voice.
Is under poverty, and there's a high number of recreation and tourism areas. Coron, on the other side, is a proportion is urban mangroves, and tourism is the leading livelihood. Since our time is limited, I just want to go straight in some of the categories that we measured well-being. And so these categories include a sense of place, where we interviewed respondents asking them if seagrass or mangroves separately, they felt connected to them. The others are memories and identity. Another category was transformative values, which respondents answered if they miss them, seagrass and mangroves, when they are away. Another category of well-being was knowledge from nature. Additional categories included social bonds, so spending time with friends and family. There was also participation, which is caretaking for the seagrass and mangroves. Aesthetics, untouched by the beauty. Health, feeling more healthy with interactions with seagrass and mangroves. And bequest values. So out of the 123 respondents, we found that sense of place, knowledge, and bequest values scored highest, and that mangrove scores were actually greater than seagrass scores. And this is on a Likert scale, 1 out of 5, 1 being low and 5 being strong and high. We also found that Philippine well-being scores are greater than Indonesian well-being scores, and this could be related to the particular context. Sorry, due to lack of time, I'll just give a quick brief summary of the in-depth interviews, where 81 respondents, or more than half of those interviews, had in-depth discussions of around an hour. And we grouped these responses in 7 themes. And actually, 2 of these themes had to do with some sort of provisioning from seagrass mangroves due to income or nutrition, while the other 5 themes had to do with cultural well-being, such as social relations, memories, beauty, skill and pride related to harvesting from seagrass and mangroves, and protection. And to conclude, one of our respondents said this, that you can't pay for the happiness we feel together when we are in seagrass and mangroves. And so what this study showed, this case study, was that these well-being scores were overall high. They were highest among resource users and lowest among the tourism sector. And we thank all our assistants and the funders for this research. Thank you.
Thank you, Frida and Angela, for the really unique and interesting talk, how to evaluate human well-beings in coastal communities in the Philippines and Indonesia. I think we have 1 or 2, we can accept 1 or 2 quick questions. If you have, please raise your hands. Okay, if you have any questions, again, please write in the chat box, and during the authors can reply during our session. Okay, thank you again, Frida and Angela, for a nice talk. And now we move on to the final speaker of this session is Professor Miguel Fortes from the Philippines. I think most of the researchers, especially on seagrass and mangrove, knows him well, but he's really leading this field of science. So we like to welcome Professor Fortes for the presentation. Okay, you can start.
Thank you, Masa. Now I'm trying to share my screen and see if I am successful here.
If not, we will take on. So please first try.
Do you see my slide? No.
Not yet.
Not yet. Okay. One more.
Maybe wait for a few seconds.
One more try, and I will request you to really operate my slide.
All right.
It's really unfortunate with my system. Huh?
Maybe I will prepare. Wait a second.
Okay, so. Not yet. Do you see it? Not yet.
Excuse me, I'm now clicking. Okay, no. It's now coming.
It's not.
Great. It's coming. It's
here now. Do you see it now?
Yes, good.
Okay, so you hear me and you see my slide. Thank
Yes,
you very
so
much.
you can start. Yes,
Okay,
good.
thank you very much for that. Now, this is my topic. Of course, community of practice, a platform to link science, policy, and practice for resilience at multiple scales. Now, here's the conceptual framework for how I'm going to approach this problem. First, we have the community of practice, and I will define it in the next slide. But that community of practice has a set of activities that address this, what I call the big disconnect between the science, policy, praxis chain. That is the middle figure. And from there, the outcome is really supposed to address ecological resilience. And then if there is resilience that is attained, that feeds back to the community of practice. So it's a very simple framework. Now let's see. This is how it is defined. The community of practice, or COP, especially by Etienne and Beverly Wenger Trainer. A group of people who share a common or a passion for something they do and learn how to do it better as they interact regularly. But please know the components of the COP. First, uppermost is the domain or the area of shared inquiry. And then right, community members, for those people committed to a process of collective learning. And then down yellow, you have the practice, investigation of key questions and gaps. And then left, continuous improvement. This is very important. It's a reflection on practice, evaluation of impacts and outcomes, ongoing inquiry, refinement of practice and methods. So that makes up the community of practice. Now, initially, in our global project on seagrass, I came up with an indicative locations of the initial 61 COPs, which I superimposed over the map of global seagrass distribution, just to say if there's any relation. But I think it's too early to say if there is. Now, 32 of these have clear seagrass science to policy mandates at different levels of governance. Now, question is, how does the flow of data and information to policy proceed? First step is kin. Inclusively come up with kin and make it easily understandable and acceptable by the people. Now, this figure looks intimidating, but actually it's simple. Let's focus on those three concentric circles at the very middle. You have knowledge gap, weak law enforcement, and ineffective governance. These are the causal factors, the root causes of the issues, which are the double ring surrounding these three small circles. So you're having issues like coastal erosion, poor water quality, resources overexploitation, biodiversity loss, and habitat degradation. Now, outermost, you have those rectangles, which are actually the technical data and the social data that our projects and our partners have generated since 2015 in this area we call Boracay. It's a, some of you know Boracay as a prime tourism destination. Now, what this kin means is that for each of those issues, there are root causes, and what scientists or social scientists and partners, stakeholders should do is to generate data information that could be useful to address each of these. The next step is dialogue. We should be regular, we should be constant. And kin should be the main topic of the frequent dialogues. In the figure, you see those round things which are actually the stakeholders, but more important would be those arrows. The thick green double arrows indicate more adequate interaction among those concerned stakeholder, but the orange double arrows, thinner ones, indicate inadequate interaction. The desired action is in blue, which should come up with informed decision, which is now the product of the discussion or exchange of ideas, information among the stakeholders, which should trickle down to the community. And the community is the one that really needs most the outcome. See, so there is really this, what I call the big disconnect, especially in terms of dialogues among the stakeholders. The next step is adaptive management cycle, which I mean here that the outcomes of the dialogue should go into the adaptive management cycle. If you look at the figure, the leftmost figure really is the constant stakeholder dialogue, which we saw in the previous slide, but the outcome should be inputted into this adaptive management cycle. Constant dialogue among the stakeholders identifies the problems, initiating the iterative adaptive management cycle until the problems are resolved or at least ventilated. I would like to emphasize iterated because this is no stopping. You have to iterate it up to the point that you really understand and solve the issue at hand. Next is, of course, from adaptive management, you try to go to resilience-based governance. Resilience-based governance incorporates the formal institutions, informal groups or networks, and individuals at multiple scales. Underline. This is for purposes of collaborative environmental management. In my particular example, I am matching agencies to their legal governance scale toward resilience of seagrass ecosystems. This is just an example. So topmost, number one, international global, UNEP, because it is mandate internationally. Down below, then you have the regional, which is in this case, the ASEAN, or the Association of Southeast Asian Nations. And then down at the national level, Philippines, local is provincial, is Puerto Princesa, and then the community, which is the Tubataha Reef. So this is really matching agencies to their legal governance scale. Or you may want to match the agencies to their geographic scales of governance. This time it's geography. So international, regional, national, and community agencies are linked to their scales of governance with an intermediary or the bridging organizations as the vehicle to fill gaps in scale matching and to facilitate communication and information flow. Again, in this example, this is environmental governance of blue carbon ecosystems in Busuanga. You have heard from the previous speaker. Busuanga is a municipality of the province of Palawan in the Philippines. In this hierarchy, the world is at the top of the scale, followed by the Coral Triangle, and then the Philippines, and then the province of Palawan, and the smallest ecological scale is Busuanga. So note the bridging organizations, the figure, international, regional, national, but very specific, especially in the Philippines, is that we have community organizations which have practically sole jurisdiction over their community resources. And then the fifth step, policy or decision-making towards resilience. Please look at the right. We are dealing here with key governance bodies which come up with activities like issue identification, then policy formulation, decision-making, implementation, and evaluation. This is a very difficult situation because most cases, what it lacks really is a completeness or inclusivity. In any case, in a policy-making or decision-making process, the main elements to consider, if you look at the ones that are in here, one, understanding of risk and drivers of change processes, social ecological systems as complex adaptive systems characterized by cycles and uncertainty, social systems and ecosystems as coupled and co-evolving, and lastly, resilience efforts must be coordinated and executed at the asset, neighborhood, or individual level. I'm showing this because I'd like to demonstrate how at the COP level, there is linking of science, policy, and practice. A case study of the Fiskil in Bolinao, that is a municipality, northwestern part of the Philippines. There are practically two parts. The one at the left is the science. The one at the right is policy and practice. With science, we start with perception survey through citizen science and satellite imagery. So that's social and technical. But what those efforts did was to produce results like there were too many fish structures in the channel where those fish cages are, and also mangroves area were declining, seagrass area were declining. And then finally, these conditions brought an increase in the residence time of sediments and fish feeds, which eventually decimated seagrass and other oxygen providers, finally giving rise to anoxia, very, very small amount of oxygen. And of course, the fish kill, a massive fish kill. That fish kill resulted in the loss of about ₱1 million a day because of, the revenue that was coming from the fish cages were no more coming. And so meetings with stakeholders come up with lessons like fish structures should be in areas where water is easily renewed, do not block channels of waterways, effort should be a considered one, and seagrass and mangroves help a lot. From these lessons, the communities decided 2.7 kilometers of fish cage areas had to be cleared from the channel. And the action was, they really came up with a MOA to clear the channel to replant seagrass and mangroves. So it's very clear, it's simple, but it's very clear how science can be very useful in linking it with policy and practice at various levels of governance. These are takeaways. There are critical barriers inhibiting knowledge exchange among stakeholders. Options for overcoming these barriers are needed. Increasing the influence and impact of science on policy and practice necessitates moving beyond the ongoing diagnosis of challenges and barriers towards a systematic study of bright spots. This will help to identify the key principles underpinning success and allow for the development of more effective strategies for successfully navigating the interface of science, policy, and practice. A willful understanding of the science, policy, practice interface at various scales is necessary to attain resilience, sound consideration of the ecological processes underpinning ecosystem services, the involvement of agencies at multi-scales, the development of projects that demonstrate not only the ecological, but also the economic and social benefits of maintaining the provision of ecosystem services, and the existence of market mechanisms might be fundamental pieces in the integration of the services into policy and practice. As my last slide, I'd like you to share with you the definition of resilience from Nelson Mandela. The greatest glory in living lies not in never falling, but in rising every time we fall. Thank you.
Okay. Thank you very much, Professor Fortes. Unfortunately, I missed the last 30 seconds. My Zoom was disconnected, but I hope everybody else could listen to the end of his talk, I hope. But anyway, that's okay. And I think we can have, as Professor Fortes' talk is quite general, we can move to the general discussion already.
Yes, of course, of course.
Okay, so first, any question to Professor Fortes or other speakers from the floor? It's okay. Okay, then let's go. There's 5 more minutes to until the end of this session, so we can discuss something about how to proceed our program, Marine Life 2030. So can I share the screen again? All right, I hope you can see my screen. So there's some tips for the discussion. First, first, Marine Life 20 has just started this year, and it's so far as you see, most of the speakers are getting their fund by themselves for the small-scale research, but not the whole Marine Life 2030. It's too big to get one grant to support everyone. So it is a kind of, it is not like a research project itself, but a collection of the different research project. I hope it is integrated. One project has to be better to be integrated with others. And so we need to make a network of research program, and this big umbrella. That is our first intention. And we have discussion. First, there's many people engaged, even from Asia. There's a lot of different researchers and different organizations, and there's a lot more in the States, US, and America, and Europe, and Africa. So communication is really important, especially not only within ourselves, researchers, but with partners, stakeholders. And so far, there's a step-by-step achievement we could do by there's several already existing upcoming events already planning, or it's actually, there's a first ocean science meeting will be held in Hawaii next February. And then there's another one, Puerto Rico. It is for the US. But there's a lot of other activities. It is US or Europe. And in our area, especially in Asia, in 2023, we have a next World Conference of Marine Biodiversity hosted by Dr. Eileen Tan. So it is a nice occasion to start discussion to how to put any achievement or output for the first few years. And we will have some discussed in the middle part of our discussion about the technologies and standard. And this time we
This is Tadashi Kajita speaking. I think unfortunately Masa Nakaoka was disconnected. So I'm now sharing his presentation. I think this is the one he wanted to show you. So anyway,
now I'm coming back. Sorry.
Right.
I don't know how far we went, but I thank you, Dr. Kajita, to take over the lead. So,
no problem.
Okay, so these are the things we need to discuss. I think we are now running out of time. I'm sorry, but let's see. We need to finish exactly by 10:30. Is it okay to continue? If you have any further questions. Anyway, we can share this discussion slide later. So any of you who are willing to participate in this project can just contact me or any other US representative, is Frank Mike Mueller-Kaga or Gabriel or anybody, but you can contact me directly if you are interested in joining this program of the UN Decade of Ocean Sciences. Okay, sorry about the interruption, but I think we have at most more than 80 speakers, 80 audience for this session. So I think it's quite successful. So I hope we can keep on discussion and contacting to each other for the further discussion and launching our interesting research topics and the collaboration, be not only research, but collaboration with stakeholders and citizens. Okay, now it is the end of our session, and thank you very much for the coming, especially for speakers to give very interesting talks. Hope we can see each other in again soon, maybe within this meeting again. Okay.
Thank you.
Bye,
Thank you,
everyone.
Masa. Thank you, Masa.
Thank you, everybody. Bye-bye.
Bye-bye.
Thank you, Sir.
Thank you.
Thank you.
Thank you very much.
Thank you very much, everybody.
Very much.
Thank you very much.
You.
Bye.